All right, in this little video, let's discuss an important complication that arises due to the use of antimicrobial drugs, and those are superinfections. So previously we've talked about how microbes can become resistant to antimicrobial drugs, and that's a big problem in the medical industry. But superinfection is a slightly different complication that arises due to the prescription of antimicrobial drugs. Now, to understand superinfection, let's first make sure that we are clear on the microbiome. So microbiome, if you search the internet for a definition of microbiome, you'll find this. The microbiome is just a word that refers to the organisms in a particular environment. In our class, when we use the term microbiome, often we're talking about the body. So the organisms in a particular environment, including the body or a part of the body. And for this video, let's just think about the microbiome as being all of the microbes that exist on the human body or in the human body. And there are a lot of them. So people have been quite interested in the microbiome and they've been studying it for years. So here's a chart that represents some of the diversity of microbes, where you find them on the body and which ones you find in different places on the body. I'm going to just show you the image to demonstrate that, again, the microbiome is very complex. Our body contains lots of different microorganisms. And we refer to them as the microbiome altogether. The other thing we should recognize about the microbiome is that with so many microbes in and on the body, some of these can be dangerous. So when they're in this normal proportion, in this balanced population with all these other microbes, they're not harmful. But some of them, if they end up in a different part of the body or if they're allowed to overgrow to a larger population size, they can become dangerous. Right? And if we understand that the human body has a microbiome, that there are lots of different kinds of microorganisms living in and on your body, and that a few of them could be dangerous if they overgrew, then we're completely ready to understand what a superinfection is. All right, so let's discuss a superinfection. The first detail we need to make sure is clear for a superinfection is how selective toxicity works. So we've talked about selective toxicity in the past. This just says that drugs, antimicrobial drugs, are highly effective against the microbe, but have minimal to no toxicity to humans, right? So you and I can take penicillin and it will harm bacteria in our body, but it will not harm us because there is no peptidoglycan in our cells to damage, right? So that's selective toxicity. The drug harms bacteria, but not human cells. Now, what's important to recognize about that is that selective toxicity does not protect beneficial microbes from harm. So again, your body is filled with lots and lots of different kinds of microorganisms. Some of those, or most of those, are beneficial. Some of those beneficial microorganisms will undoubtedly be harmed if you take penicillin. Hopefully the penicillin will kill the organism that is making you sick, but again, undoubtedly some beneficial microbes will also be killed. So the takeaway message there is antimicrobial drugs harm or destroy both harmful and beneficial species of microorganisms in your body when you take them. All right. So that tees us up for, well, what's a superinfection? So what is a superinfection? So we give a patient an antimicrobial drug to treat an infection. If everything goes according to plan, that drug kills the microbe causing the infection. So that's great. That was our goal from the outset. So again, let's think about penicillin. We prescribe a patient penicillin. It kills presumably, most likely, a gram-positive microbe that is making them sick. But just like we said a moment ago, penicillin is not designed to kill only the microbe that is making a person sick. So unfortunately, the penicillin also kills beneficial bacteria. And this will be true for every single antimicrobial drug. Hopefully it will kill the microbe that is making us sick, but hopefully it will also, or unfortunately, it will also kill microbes that help keep us healthy, beneficial ones. So, in the absence of these beneficial microbes, some microbes that were once a small part of the microbiome can overgrow and cause disease. Again, our microbiome is filled with lots and lots of different microorganisms, many of them in very small populations. But when we throw that population out of balance by removing one of the beneficial microorganisms, it frees up space or habitat for these less desirable microbes to overgrow into. And that can lead to a second disease, right? We treated this infection with penicillin, but unfortunately the treatment allowed another bacteria to overgrow, causing disease. Again, these harmful microbes are usually kept in check by the beneficial bacteria. Unfortunately, we accidentally killed those beneficial bacteria. So again, this is a superinfection. Let's look at a superinfection visually. So here we've got an infection in the throat, strep throat, let's say. We do aggressively treat strep throat because of the dangers of that infection. So we give a strong antibiotic, trying to kill the organism that is causing that throat infection. So we take the drug, but the drug does not stay just in the throat. The drug circulates through the entire body. Hopefully the drug kills the microorganism causing the disease. So this patient now has a not sore throat, right? So we've cured that infection. But if we think about another place in the body in this patient that is being treated with our drug, like the intestine, we can see that the drug is also active in the intestine, killing microorganisms there. So in this simple intestine model, we've got a population of bacteria that's mostly triangles, and these triangles are beneficial bacteria. There's a couple squares. There's just a couple. If these squares overgrew, they could be harmful, right? So again, we take the drug. Unfortunately, it kills some of our beneficial bacteria, freeing up resources in this habitat, and then the squares overgrow. And this person who once had a sore throat might now have gastrointestinal disease because of the drug treatment, making this a superinfection. So let's talk about two really quick examples of superinfections that can commonly occur. All right, so two examples. The first one is when we treat urinary tract infections. So urinary tract infections are extraordinarily common. We might treat a UTI with a broad spectrum antibiotic, let's say cephalosporin, okay? And we're treating a UTI caused by E. coli. So cephalosporin will kill E. coli really well. This is perfect. Our drug treatment cures this UTI infection. But we have to remember that every single time we use an antibiotic drug, hopefully we'll kill the organism causing a disease, but unfortunately we'll also kill some beneficial bacteria. So the problem with this treatment is that this drug will also kill a form of bacteria that is a normal inhabitant of the vagina. And this bacteria is called lactobacilli. Lactobacilli, again, normally live in the vagina. Lactobacilli do tons of lactose fermentation. That's where their name comes from, the lacto part. We know that fermentation creates acid waste products. And so these bacteria that are normal inhabitants of the vagina consume sugars via fermentation, creating acid waste products, and they actually maintain the normal pH of the vagina to be slightly acidic. So a healthy vagina environment has an acidic pH. But again, we took this drug to treat this UTI, and unfortunately, sometimes these drugs will also kill the lactobacilli in the vagina. That means the lactobacilli are not there to create acid waste products, and the pH becomes more neutral. When that happens, another microorganism called Candida albicans overgrows. Candida doesn't like acidic conditions. It's a neutrophile. So when the pH of the vagina changes and becomes more neutral or less acidic, Candida albicans then goes crazy and overgrows, causing a yeast infection. So Candida albicans is yeast, and a common superinfection is a yeast infection that follows a UTI treatment with a broad-spectrum antibiotic. One might ask, hey, why doesn't the cephalosporin kill Candida albicans? And I'll leave you to think about that and try to answer that for yourself, and I hope if you are struggling with that answer, you'll let me know. So let's think about one other example. So a second example of a superinfection that we might commonly observe in the world. All right. This is when we use an antibiotic drug and we end up with this antibiotic-associated colitis, often caused by a bacteria called Clostridium difficile, or you might recognize the name C. diff more commonly. So this is going to be more common for, let's say, anybody that's in a hospital with some sort of infection. So we're giving antibiotic treatments for any number of reasons. So in this example, our patient is just plain ill, perhaps with an infection, and we prescribe a drug, an antibiotic drug, to try to help the patient. Unfortunately, they end up with this superinfection called colitis. So what happens, right? So we give the drug to treat whatever infection this patient has, but again, and hopefully it works, but again, unfortunately, every single time we prescribe an antimicrobial drug, not only do we kill the bacteria causing us harm, we also kill some other microbes. So we kill some other microbes, and then this bacteria, Clostridium difficile, or C. diff, overgrows. More specifically, antimicrobial drugs tend to kill a bunch of bacteria in our intestines. Our intestines are the largest habitat for our microbiome. So there's lots of microbes in our intestines. Whenever we take an antimicrobial drug, we undoubtedly kill some of the microbes in our intestines. Again, that frees up space, and Clostridium difficile overgrows. Clostridium difficile does tons of damage to the intestines, causing inflammation, which leads to diarrhea. Clostridium C. diff diarrhea is particularly awful. So again, the reason this happened is our patient was ill, we prescribed an antimicrobial drug, hopefully that cured the infection. Unfortunately, it also removed some of the beneficial normal microbes from our intestines. C. diff then overgrows in our intestines, leading to intestinal damage and severe diarrhea. So we might ask a similar question about Clostridium difficile. Why doesn't a broad-spectrum antibiotic kill C. diff? And Clostridium is one of about 3 genus of microbes that have a unique characteristic that protects them. Clostridium is protected because they can form spores, endospores. And that endospore form is resistant to drug treatment. So while the patient is ingesting the antimicrobial drug, Clostridium difficile is not killed because it can be in the endospore form. As soon as the drug clears the system, and there's all this extra space in the intestines because we killed a bunch of microbes, Clostridium difficile endospores can germinate and immediately start growing. They overpopulate that space, they cause damage to the intestines, and they stimulate diarrhea for these patients. So those are 2 common examples of superinfections in our everyday world. As always, if you have questions about superinfections, I hope you'll let me know. Otherwise, I'll talk